Okay, can you hear me okay? All right, should we get started? Okay, welcome. So the agenda for today, we're going to wrap up our discussion of folate, vitamin B12, and S-adenosylmethionine metabolism in the first hour. And then we're going to switch to nucleotide metabolism for a couple hours. So it'll be a marathon, but well, I guess it'll just be a marathon. Are there questions from yesterday's session? Yes.
So there was a slide that talked about, I think, like 5 different NM configurations of the tetrahydrofolate. Is there any particular ones that are most relevant as far as like the most reduced form and the non-reduced form, or should we try to commit to memory all those different versions?
So there's 4 forms that you should know. The N5 formal as the most oxidized. The 510 methylene, 510 methanol, and then the methyl tetrahydrofolate. So those are the major forms that are used for carbon donation reactions. So yeah, I think you should know those. Other questions? Did anyone get a chance to review this figure? Is it helpful or obfuscational? Not sure if that's a word. Okay, so this is a summary that is a little hard to parse out at the first glance, but I think it's pretty useful. So over here, this is the thymidine synthase side. Okay, so thymidine synthase is right here. That leaves the folate in the dihydrofolate oxidation state, and then it has to get re-reduced to tetrahydrofolate. Then the methionine cycle is shown down here. Okay, the donors are shown up here, and the different forms of tetrahydrofolates that we get when we get carbon donated from different sources. Okay, just to preview something that we haven't talked about yet, but we will talk about in the second and third hours. This formal tetrahydrofolate, this is a carbon donor for the synthesis of purine nucleotides, or for the base portion of the purine nucleotides. Okay, so this figure, again, it's a little hard to figure out what's going on if you just look at this in isolation. But if you have this figure out while you're reviewing some of the other material, I think it's pretty useful. Were there questions about this? Okay, so we're going to talk about some different drugs that affect folate metabolism. And we're going to zoom in on this side, the left side of the pathway, and that thymidine synthase reaction. Okay, so thymidine synthase, the enzymatic function is to convert deoxyuridine monophosphate to deoxythymidine monophosphate. And the difference between the uridine base and the thymidine base is the presence of this methyl group. Okay, so if methyl tetrahydrofolate was a donor, then we'd have a methyl carbon here and a methyl carbon here, and that would be the end of it, right? But we don't have the methyl carbon as the donor. We have the methylene carbon as the donor. So in order to make this a methyl, we have to strip some electrons from the tetrahydrofolate itself. Okay, so when we do that, then we're left with the dihydrofolate. That has to be re-reduced by dihydrofolate reductase to tetrahydrofolate. Is that clear? We talked about this yesterday. Okay, so let's look at some drugs that will target this pathway. And why would we want to target this pathway? Well, rapidly dividing cells like cancer cells have a high requirement for replicating their genomes, and they have a high requirement for nucleotides. Okay, so if we can starve these cells of their nucleotide pool, then we can slow their cell division, and that's beneficial for cancer. Okay. Also, inflammation, that requires cell division as well. You've got to have lymphocytes and et cetera proliferating. So these drugs have or methotrexate at least has a role in inflammatory diseases like arthritis. Okay, so let's look at methotrexate first. Methotrexate is a competitive inhibitor of dihydrofolate reductase. And if you look at the structure of tetrahydrofolate here, methotrexate, the structure is almost identical. So the differences, so the methotrexate has a methyl where methyl tetrahydrofolate would be. So it's kind of resembling methyl tetrahydrofolate. And then instead of an alcohol at this position on the pteridine ring, we have an amine group. Otherwise, it's structurally identical to the normal substrates of dihydrofolate reductase. So the methotrexate, this has roles in anti-cancer treatment and in treatment of inflammatory conditions like arthritis. 5-fluorouracil, this is a drug that I think this is only used in malignancies. I don't think this has a role in inflammatory diseases as methotrexate does. But 5-fluorouracil, this is acting, it's acting as a prodrug, meaning that it has to be modified by cellular enzymes before it can be an active drug or an active enzyme inhibitor. And the pathways by which it is activated, we'll talk about some of these in our discussion of nucleotide metabolism later, but essentially the 5-fluorouracil, this is an analog of the base, the uridine base, the uracil base. The 5-fluorouracil has to get modified by the addition of a ribose sugar and then a phosphate group to make it into a nucleotide. So the prodrug is an analog of a base, and then it gets converted into a nucleotide by the same intracellular enzymes that would do normal nucleotide metabolism. Okay, so the nucleotide version of the fluorodeoxyuridine, this is acting as a suicide inhibitor of the thymidine synthase enzyme. So it's making a covalent bond with the cysteine side chain in the thymidine synthase active site. So the drug is shown down here. This structure up here, this is the methylene tetrahydrofolate, and this is the carbon that the thymidine synthase is trying to abstract and make into a methyl carbon to convert the uracil into a thymidine. Okay, the presence of the fluorine atom here, that's preventing the catalysis and is essentially locking the enzyme in this position with a covalent bond to the drug. Okay, so this is acting as a suicide inhibitor and is permanently knocking out the enzymatic activity of the thymidine synthase. All clear? Okay, should we do a clicker question or two? And these will be pretty easy, but imagine this is this time next week and see if this will stick. Everybody get a chance to answer here. Ooh, very good. 100%. Very well done.
Final, right?
Final's getting filled up. Okay, so you have to know what enzyme is inhibited and what the product of that enzyme is, right? Okay, so the product is deoxythymidine monophosphate. The substrate is deoxyuridine monophosphate. So we would expect there would be an accumulation of the deoxyuridine monophosphate and a depletion of the deoxythymidine. All right, and you probably knew this was coming, right? Blast from the past. Are you ready? Ooh. Best answer here is Vmax is decreased. So this can't be a competitive inhibitor because the drug is making a covalent bond at the active site. Right, so you can't just dump on more of the deoxyuridine monophosphate and outcompete it because it's covalently bonded there. Okay, so the kinetics of a suicide inhibitor are similar to the kinetics of a non-competitive inhibitor with a decrease in Vmax. Questions on that one? Okay. All right, so let's move on and talk about some things that can go wrong with folate metabolism and vitamin B12 metabolism. So there is a common phenotype that can result from deficiencies in folic acid and deficiencies in vitamin B12. And this condition is an anemia with larger than normal cells. So this is referred to as a macrocytic anemia, so fewer than normal red blood cells, and the red blood cells that are there are larger. So macrocytic anemia is not exactly synonymous with megaloblastic anemia, but they're related. So the megaloblastic anemia, this is referring to the presence of blast cells or immature red blood cells that are released from the bone marrow as a compensation for the anemia. Okay, so they're trying to pump out immature red blood cells. So you'll see these red blood cells that have a nuclei. And the rationale for, or the mechanism by which the macrocytic, yes?
Do we consider them to be similar because you talked earlier, like yesterday, when you'd say the sign for folate is macrocytic anemia, and you'd be saying that, and on your slides a lot it would say like megaloblastic. So do we think of it as the same?
I think it's synonymous.
Synonymous? You would never have like a multiple choice with both options. So if you
I would never know. I mean, a hematologist might, right? But that's a finer distinction that I would make.
Okay.
So I think you can have a macrocytic anemia without the blasts, and then if it gets a little bit worse, then it becomes a blastic anemia.
Okay.
But yeah, the fine points of distinction between the different anemias, save that for immunology, hematology, oncology in the spring. But just know that the most important thing is to understand the mechanism. So the red blood cell during its development, it has to do, essentially two things. It's got to increase mass, and the mass comes mainly from hemoglobin. And then it's also got to replicate its genome, right? And the genome replication, that requires nucleotides, And if we don't have folate around, we've just saw with the thymidine synthase reaction, if we don't have folate around, we can't sustain the deoxynucleotide pool that's necessary for cell division. So the macrocytic anemia that results from a folate deficiency or a vitamin B12 deficiency, the idea here is that the red blood cells are building mass. They don't have any problem with hemoglobin synthesis or iron metabolism or anything like that. So they're building mass, but that division of their genomes that's required in erythropoiesis, that is delayed because of the paucity of the deoxynucleotides. Okay, so you get a larger than normal cell because it's still able to pack on the hemoglobin, but the division of the cell is not coupled to the cell's increase in mass as it normally would. Does that make sense? No? Okay. Okay, so with a folate deficiency or with a vitamin B12 deficiency that would give you a functional folate deficiency, you have a lack of nucleotides, a lack of deoxynucleotides. Okay, so a red blood cell or a red blood cell precursor that's in the bone marrow, it's trying to divide, but it can't divide because it doesn't have enough nucleotides to do it. Okay, but it can still synthesize hemoglobin and still pack on mass. So it's getting bigger and bigger, but you know, it gets bigger, or it gets to the size where a normal cell would divide, but it can't divide because it hasn't replicated its genome because the nucleotides are limiting. Does that make sense? Now that's in contrast to like an iron deficiency anemia. Iron deficiency anemia, the problem is exactly reversed there. That results in a microcytic anemia, right? So there, with the lack of iron, you don't have a problem with nucleotide synthesis. Those are plentiful, but there, with the lack of iron, you don't have hemoglobin. You can't make hemoglobin. So there it's like the exact opposite problem. The cell can replicate its genome just fine, but it can't build mass. Okay, so kind of, you know, we'll talk about iron homeostasis in IHO and elaborate on that then, but useful to keep those two ideas in your head. Okay, let's see, got off on a tangent here. Any other questions? I do have a question for you. Are we ready? Ooh, almost 100%. Good. So elevated homocysteine. So this is a condition sometimes referred to as hyperhomocystinemia, or elevated homocysteine in the blood. It's also sometimes called homocystinuria or urinating out homocysteine. And this is associated with vitamin B12 deficiency, and it can also be associated with loss of function mutations in the methionine cycle. So essentially with the hyperhomocystinemia, the lack of the vitamin B12 there, the methylcobalamin that is required by methionine synthase is absent, so you don't get synthesis of methionine and you get a homocysteine accumulating. So the pathology of the hyperhomocystinemia, it's associated with a lot of cardiovascular problems, like if you just take a random sample of people's homocysteine levels, it's associated with kind of chronic conditions, but the mechanistic link isn't so clear. And it may be that the homocysteine itself isn't so toxic, but it's really reflecting rather a lack of the methionine cycling and lack of S-adenosylmethionine available for methylation reactions as a methyl donor. Now, if somebody has hyperhomocystinemia as a result of vitamin B12 deficiency or because of a loss of function mutation in methionine synthase, for example, there are other routes for homocysteine metabolism. The homocysteine can be methylated by using a different methyl donor that's derived from choline called betaine. And this betaine, this is like a trimethylated glycine. So this can be an alternative methyl donor in the absence of vitamin B12 or in the absence of methionine synthase to convert the homocysteine into methionine. Homocysteine can also be catabolized into cysteine. This is through vitamin B6 dependent pathways. If you have a vitamin B6 deficiency, that can also manifest as a hyperhomocystinemia because this pathway of homocysteine catabolism is absent. Okay, so let's look at that betaine pathway. There is a molecule called choline, and choline, we may have heard this in phosphatidylcholine as a part of an important membrane phospholipid. So the basic structure, you've got the 2-carbon ethanol here. Then we add an amine group to it. That would be ethanolamine. And then we add a methyl group or 3 methyl groups to the amine group, and then that becomes choline. So this alcohol, this can get oxidized to an aldehyde and then to a carboxylic acid, and that's the betaine molecule. Okay, so betaine, that's got these nice 3 available methyl groups, and betaine can donate one of them to homocysteine to make methionine as an alternative route of making methionine in the absence of vitamin B12 or in the absence of methionine synthase activity. So betaine, this is a naturally occurring molecule, but it can also be a treatment for the hyperhomocystinemia. This pathway is not present or so this pathway is less active in neuronal tissues, and that makes them susceptible to the hyperhomocystinemia. Or, excuse me, maybe it's more accurate to say it makes them more sensitive to a lack of methionine synthesis. Okay, so if that methionine synthase activity is corrupted either because of a lack of vitamin B12 or due to an inherited mutation or due to a folate deficiency, for example, the homocysteine will build up, and this buildup can have effects on neuronal tissue and also on bone formation and also on vascular metabolism. So these effects on the peripheral and central nervous system, it's not really clear whether this is because of homocysteine toxicity or because of lack of methionine and lack of methionine signaling. The bone metabolism, homocysteine is interfering with the collagen maturation reactions that are necessary for generation of collagen and mineralization of collagen that are how bone is made. Okay, sorry. Okay, so we could get this hyperhomocystinemia as a result of a folate deficiency, as a result of a B12 deficiency, or as a result of a loss of function mutation, for example, in the methionine synthase. Okay. So just as a okay, so this is pretty, easy question now because we just said it. 97%, good. So beta-ene, the idea in the lack of vitamin B12 here is that we can't do this reaction. So if we can supply beta-ene, that can allow methionine to be synthesized from homocysteine through an alternative pathway. Okay. Now, let's see. Most of what we've talked about with the lack of vitamin B12, and if we have a lack of folate, we're going to have a functional lack of B12, right? Because we can't get the methyl B12. So the methyl B12 is an important part of the pathology of vitamin B12 deficiency, but it's not the only part. Remember that there's another reaction that vitamin B12 plays a part in, and that is as adenosylcobalamin for the methylmalonyl-CoA mutase reaction. Remember that one? So the methylmalonyl-CoA, if you'll recall, this is a product of the catabolism of branched chain fatty acids and odd chain fatty acids and some of the branched chain amino acids. And so we make methylmalonyl-CoA, and then the methylmalonyl-CoA mutase reaction, which requires adenosylcobalamin as a cofactor. This unbranches it and makes it into succinyl-CoA. Okay, so if we have plenty of vitamin B12 around, this is happening, and then something that we're going to talk about on Friday is also happening, fatty acid synthesis. So fatty acid synthesis, just to give you a 2-second preview of it, fatty acid synthesis uses malonyl-CoA as a substrate. So malonyl-CoA is a 4-carbon straight chain carboxylic acid. Okay, and when we use this as a substrate for fatty acid synthesis, we make straight chain fatty acids. Okay, no branch points, and this lets us make phospholipids that pack nicely together and form a membrane that has good membrane integrity. Now, if we lack if we lack adenosyl B12, the methylmalonyl-CoA, then this doesn't have a metabolic fate. This accumulates, and the fatty acid synthase enzyme will use this inappropriately as a substrate. That will result in branched chain fatty acids being synthesized. Those then are incorporated into phospholipids, and they can't pack together as nicely because of the branch points, and that leads to poor membrane integrity. And this is, again, particularly important in neuronal tissue, in central nervous system and the peripheral nervous tissue. Okay, is that clear? And this part about the fatty acid synthesis, we'll spend a couple hours talking about that on Friday, but just as a preview there. Okay. Shall we move on? Okay. Ooh, very good. Okay, so from yesterday's lecture, we know that this final reduction step to make methyl tetrahydrofolate, that's irreversible. Right, so that if we have methanyl tetrahydrofolate, for example, we can reduce that to the 5,10-methylene, or oxidize it to the 10-formyl, so we can freely flip back and forth between these three forms. But when the 5,10-methylene tetrahydrofolate is reduced to the 5-methyl tetrahydrofolate, that then is an irreversible reaction. Okay, and this is important because this is helping to explain some of the pathology of a vitamin B12 a vitamin B12 deficiency, and why that is recapitulating some of the features of a folate deficiency. So the methyl tetrahydrofolate, the only fate of this in our body is to donate this methyl group to cobalamin to make methylcobalamin. Okay, so there's no other reaction that this methyl tetrahydrofolate can participate in. Is that clear? Okay, so the consequence there is that if we don't have vitamin B12 in our body, if we don't have cobalamin around, all of the folate pool will get trapped in this methyl tetrahydrofolate form. Okay, because we can freely interconvert between all these forms, but then when we go to the methyl form, we're trapped there because we can't reoxidize it and we don't have cobalamin around to make methylcobalamin. Okay, so this is why the vitamin B12 deficiency will cause a functional folate deficiency and result in the macrocytic anemia. Now, vitamin B12 doesn't directly participate in the thymidine synthase reaction, right? So it doesn't directly impact the availability of the deoxythymidine nucleotide, but it indirectly impacts it because it makes all of the 5,10-methylene tetrahydrofolate. That's not available anymore. It's all of the tetrahydrofolate pool is locked up in this methyl tetrahydrofolate form. Okay, is that clear? Okay, so that's the methyl trap hypothesis, and it's why the vitamin B12 deficiency will recapitulate the macrocytic anemia of a folate deficiency. Okay, so you get this larger than normal red blood cells and the presence of blast cells as a compensation for the anemia. So this part of the vitamin B12 deficiency, this goes right along with the folate deficiency. Vitamin B12 deficiency has additional symptoms, and these are largely neurologic, and that is because of the issues with the homocysteine metabolism and that being compromised. The autoimmune gastritis, this is frequently the initiating factor for the vitamin B12 deficiency. So the autoimmune destruction of the parietal cells that are secreting intrinsic factor that's required for the vitamin B12 deficiency, or excuse me, that's required for vitamin B12 absorption, that's one of the more common causes of the vitamin B12 deficiency. So the clinical presentation, or this constellation of the macrocytic anemia with the other neurological symptoms of the B12 deficiency, together we refer to that as pernicious anemia. So it's like a folate deficiency plus extra neurological symptoms that indicate the vitamin B12 deficiency. Okay, are there questions on that? Okay. This cartoon is just kind of a summary of cobalamin metabolism and the methionine cycle that I thought was a good summary. So I'm not going to go through it all, but I'll just leave that for your review. And if you have questions on it, we can discuss them. Should we go through a case or two? Okay. Okay. Let's see. Severe megaloblastic anemia, developmental retardation, and intractable respiratory tract infection. Okay. How would you approach this patient? Do some labs?
Yeah.
Yeah. It's a good way to kill some time while you're collecting your thoughts anyways. Okay, so the patient has low hemoglobin and a low hematocrit. Right, so this is our definition of an anemia, right? And then the mean corpuscular volume, this is the average volume of a red blood cell that's higher than normal. So that's where we get our macrocytic anemia. Okay. What else is significant here? There's elevated iron, and that's probably a result of the anemia. So the major route of iron, or the major fate of iron in our body is to support erythropoiesis, and if that's not happening normally, then you're going to get elevated iron. Okay, vitamin B12 looks good. What do you think about the folate level? Yeah, it's way down. Okay, well, what would you do with this patient? I guess I should note here, I mean, I was focusing on the red blood cells, but that lack of nucleotides, that can impact other cells as well. And in this case, we've got, let's see, the lymphocytes. I guess the lymphocytes were normal here. Okay, I take it back. I was there. Okay, that's what. Thank you. So this would be a thrombocytopenia, or a low platelet count. So the megakaryocytes, those are cells again in the bone marrow that make platelets, and if they can't replicate, there's going to be a deficiency in those cells. Okay, so let's see. So she had a pneumonia, and the organism that was cultured, or that was identified in her lungs, was Pneumocystis jirovecii. Has anyone heard of that before? What's the significance of Pneumocystis jirovecii?
Pneumonia, usually?
It's a pneumonia, but it's a pneumonia that's, if you have a healthy immune system, you wouldn't get it. So it's a pneumonia that's associated with advanced HIV or immune compromised situation. Does that sound good? This used to be called Pneumocystis carni, so sometimes you'll see that still. Okay, well, what do you think then?
Why is it an issue that trimethoprim was given? Because doesn't it target some of like this pathway that we're trying to fix, or is it totally separate in that sense?
Trimethoprim sulfamethoxazole, that's going to target the bacterial dihydrofolate reductase.
Yep. Is methotrexate the
Yep, methotrexate targets the human one. Okay, so we've got immune compromised, anemic, and folate deficient. Any thoughts on that? Okay. Have we heard the word parenteral before? What does parenteral mean?
Not via the gut, or so maybe
Yep.
is it just via the blood? Maybe is it just going IV?
Yep, exactly. So the enteric system, I guess, is the gut. So this is around the gut, or around the gut, I think. So this is giving the patient folate into her arm, into her blood. And after 3 days, the anemia and the thrombocytopenia improved, as well as her pneumonia. So why do you suspect that is then?
I mean, is it, like you already said, the trimethoprim? The one with the T prevents the bacteria from stealing your folate, so you're able to get your folate levels back in check, which fixed the other stuff?
Oh, okay. I think that's a good thought, but I don't think it's that. But I think that's a good idea, though. Yeah, I hadn't thought about that.
I think maybe it has to do with like absorption in the gut of the folate.
Yes, exactly. Absorption of the gut. So she's getting plenty of folate from breast milk or formula or whatever she's eating. I can't remember if it said that, right? But she's got this anemia, and then you give her a shot of folate in the arm, and then the anemia starts to resolve, right? So that's, pretty strong evidence that she's got a problem with folate absorption. Does that track well? Okay, so how does folate get absorbed from the gut?
The PCFTs.
The proton-coupled folate transporter. And this is encoded by the SLC46A1 gene. Don't worry about remembering that. But our patient was compound heterozygous for a loss of function mutation. And did Cormier talk about compound heterozygosity? So it's like you've got one bad allele from the mom, one bad allele from the dad. But it's different mutations in each allele. Okay. All right. Now, there's kind of a little complication here in that the parenteral folate therapy, that normalized her hematological profile, like her anemia resolved and the thrombocytopenia resolved, but the folate level in her cerebral spinal fluid was still low. Okay, so presumably that same gene is involved not just in transporting the folate from the guts into the blood, but also is important for transporting the folate from the blood into the cerebral spinal fluid. Okay, so they increased the dosing basically and pushed more into her cerebral spinal fluid. Okay. Okay, is that clear there? So this was a case of an anemia and also an immunodeficiency as a result of impaired folate absorption.
Is that normal where they test the cerebrospinal fluid, or would there have been symptoms that would have been specifically result within the CNS? Or
I don't know what the rationale is for that. I mean, testing the cerebral spinal fluid is a pretty invasive procedure, so they wouldn't generally do it unless this was either a well-known phenomenon that they had to monitor, or else she had some neurological symptoms that they wanted to test. Yep. Other questions? Okay. Let's look at 100? This is a fair question, right?
Yeah.
Okay, so that's not too easy. All right, should we do one more case in the last 4 minutes? Okay. So we had severe nearsightedness, abnormalities in the skeleton, so kind of a protruding chin, and then he had the pectum excavatum, or that kind of sunken chest phenomenon. What are your impressions? Okay, well, let's look at a little bit more information. So, yeah, not a radiologist here, but these vertebrae, they should be more rectangular. And the patient's vertebrae have these kind of divots in the top and the bottom. And these are sometimes called codfish vertebrae. Okay. All right. So vitamin D was low. That's something that could cause osteoporosis like this, but not generally in a 17-year-old. Okay. Any labs that you'd like to look at?
Homocystinuria.
Homocysteine in the urine? Okay. How about homocysteine in the blood? Good thought.
Oops.
Okay, so we've got reference values on the left and then the patient values on the right. How could you have very high homocysteine and also very high methionine at the same time?
Diet.
Yeah, diet. Okay, then what else is significant? So the vitamin B12 is fine, the folic acid is fine, and he is not anemic, right? So normal hematological problems. Okay, so the patient had a, so normal folate, normal cobalamin, normal methylmalonic acid, that's a result of the methylmalonyl-CoA. So this is suggesting that the vitamin B12 is available for at least some purposes, right? In this case, the mutation, so this was, patient's condition was caused by a inherited mutation, not in the methionine cycle, but in that alternative pathway by which homocysteine can be converted into cysteine. So the cystathionine beta synthase gene, this is an alternative route of homocysteine elimination outside of the methionine synthase reaction. But this still is going to result in an increase in homocysteine and then kind of that, what you might not expect, the increase in methionine as well.
Wait, so how's the methionine getting? I'm sorry.
Well, you just
get backed up, like the whole thing?
Yeah, so normally if you would have excess homocysteine, this is like a cycle that's continuing around. And if you had too much of it, it would go out through this pathway. But it can't go out through this pathway, so we just get too much of it all throughout the cycle. Okay. So the homocysteine, that's affecting that cross-linking of collagen, and that can account for the skeletal malformations and in our patient. So The osteoporosis, that's a normal consequence of aging in most people, but it's very unusual in a 17-year-old. So that hyperhomocystinemia / homocystinuria, the clinical pearl here is that you should always look for that if you have severe osteoporosis in a young person. Okay, and this, sorry, I don't want to go into your break here, but just some more information about the outcome of the patients and doing dietary changes that can keep his homocysteine in check. Okay, why don't we take a break and we'll do a few more clicker slides and then transition to nucleotide metabolism at 2:01.
Let's roll. I just want some. Love. Perfect combo. It is. Yeah. Okay.
That's not active.
Mine always an issue when I would close my computer.
Yeah, exactly.
Okay. But every time I close my computer, why do we see
This might be way off base, but
could she not get rid of
Collagen is important for bone formation, but it's also important for a lot of other.
wait, am I driving? Wait, what? No. Because otherwise, when you're like, you can have fully,
I think that's more likely than the.
For some reason.
oh, I wasn't. I was like him, but I was like, you guys can.
I have no idea. I just chalked it up to athletic symptoms.
I was like, I got a question.
No, I already have it.
How fully, I don't think that like so.
I thought they were all going for coffee. I just kind of zoned out.
Right, and I think that's
Hydroxylation where you just have
yesterday.
I think he did.
Me either, I'm stuck.
You just need to assume these being discriminated against by these systems because they are, like I said yesterday, it's not a mono, it's not, there's not one, you can't sum up, the rich experiences of so many different cultures by like one experience, and similar to like a health condition, if you're, like let's say, it's more likely for people who live in a certain town because of the mill that's there, I don't know if you've heard, that idea that some of these mill towns have called Parkinson's. If you're a doctor in that town, like, you probably like are gonna be maybe there might be third way or experiences. Being ready to
This one's elevated, which one?
and on the lookout for to care for them. And